A very warm welcome to all of you. Let me introduce myself. I am going to start my new channel on YouTube and I will upload videos after if I get a good response and after every week I will upload one video regarding biotechnology and as I am a student of biotechnology I will upload related videos and after that I am also going to open another channel in which I will upload videos regarding government exams and especially CDS, combined defense services. Okay, so as I will study some topics I will upload videos, and after studying I will upload it. Okay, so if you like this video, uh, then share with your friends and comment and subscribe. Thank you. So let's start the topic. So my first video is on batch culture. There might be some issue regarding the video quality, so please adjust this because right now I am not having that much amount of gadgets, so I am making this video from my mobile itself. So what is batch culture? The first question arises in my mind. So, in batch culture, what is the basic principle of this batch culture is we add the substrate one time and we extract out the product finally. There will be no addition of any kind of substrate and enzyme between the processes going on. It will be a closed system and the whole reaction will take place inside the vessel. After that, what will happen if we want to know what is going on inside the batch culture, how many cells are multiplying, how many cells are utilizing the substrate, and it is going to uh, it is going to make a unique beneficial product. Then there are certain equations, or you can say derivations, that can help us to determine what is going on inside the batch culture. So suppose if we want to know about the cell culture, what is going, so the first derivation or the basic equation that helps us to determine the cell culture is, so this is the basic equation. Let me write it, equation 1. This is the basic equation to calculate what is going on inside the bird, and where mi is the mass input, mo is the mass output, rg is the rate of generation of product, and rc is the rate of consumption of substrate, m is the total mass. So there are values regarding these constants, so I am writing rg will be equal to mu xv, rc equals to kdxv, or m equals to xv. So xv, what is mu, what is x, what is v, what is kd, and xv is common in this rc and m. So what is mu? Mu is the specific growth rate, x is the biomass production, v is the volume of the total system, and kd is the specific death rate. Okay, so you can see this growth and consumption is being minus over here to calculate the net amount of what net amount of the substrate, means the organism are dying or live, they are calculated on different parameters. So what will happen, let me one another thing is mass input and mass output will be zero. Why? Because we are not adding any mass in between the system. If we are going to take out, and not this thing will happen if we are not going to take anything in between the system, so this will become zero. Now put these values in the equation. It will become like this. No, sorry, this will become xv. I'm really sorry. So this will be equation 2. So we will cancel out all these v's. Now we will going to differentiate it with respect to t. So this x with respect to t, mu x minus k dx. Okay, so we will take out common as x. So this dx dt equals to mu minus kd. This will become x. Now this x will come on this side. Dx upon x equals to mu minus kd dt. So now we will integrate it. The limit will be 0 to t, and x naught means x initial to x final, or you can say x total. So okay, so x total. Now what will happen, we have completed over here. Let's put these values in the equation. So log will be applied here, x total upon x input, x in node, mu minus kd. This will become t. Now what will we do, we will remove this log, and this will become x. Okay, no worries. Mu, we will take mu as maximum because specific growth rate is will be, we are assuming that specific growth rate is maximum, so it will become mu max minus kd t. Okay, after that we don't want to calculate the death rate, we are only concerned about the specific growth rate in all the time. So kd will be assumed 0. So don't be panic, because it's a game of only assumptions, this equation based on only assumptions. So don't be panic, it will not be going to make changes any further. So this will become 0. Now this t, now we are going to remove this e, or you can say e value. So this will come here, and 1 upon mu max ln x total upon x naught. So this is x total itself, I have written x t, so don't be panic, it's nothing from outside. Okay, this x is written over here. After that we will convert this equation into this form, x equals to x naught e mu max and t, or you can say tb. So this is the equation to calculate the growth of the cell culture inside the vessel. Okay, in this video itself I am also going to tell you about the substrate utilization during the process. This equation will be continued in this uh, derivation. So you can pause this video and you can make notes, and I advise you to carry a notebook and pen with you, so you can write these derivations on that notebook. So this next derivation is the based on substrate utilization, how much substrate will be utilized by the microorganism for the desired product formation. So in this equation we will write the basic equation. Okay, so what will happen? This will always be 0. This, in the previous equation we have written it d dx by dt. What does it mean? X is the biomass, so here we have to calculate substrate, so we will write in place of x s v upon dt. It means concentration of substrate and volume was already there in the equation. So rate of consumption, so it will become minus rsv. This will become 0. Why it is minus? Because the concentration of the substrate is decreasing, so that's why we have taken this my rs as minus. Okay, so let's do the equation. dsv upon dt equals to minus rv. Okay, s is cancelled, sorry, v is cancelled, and write this equation into a big form. So ds upon dt equals to minus rsv equals to mu upon yxs plus qp upon qpx plus ms and xv. So v will be cancelled. This is v is written. Okay, v is cancelled. Then this equation will become will be put into this format. Okay, the value of r, this is value of rs, will be put into this equation where v was cancelled. So let's put this equation. ds over dt is equals to minus mu upon what is yxs plus qp upon qpx plus ms x. Okay, so we have calculated the value of x in the equation in the previous derivation. So the value of x was x naught e to the power mu max tb. We will put this value of x into this place. Okay, so ds over dt equals to minus mu upon yxs plus qp upon qpx plus ms x naught e to the power mu max tb. Okay, so now what will we do? We will remove this part. You know how to remove this log. You have studied in semester 2 and 1. So tb will come here, 1 upon mu max will be here, and ln 1 plus s naught minus sf upon mu upon yx. This is mu max. Okay, yxx. Okay, this is mu max. Okay, this is not mu max plus qp upon qpx. This will be mu max again, mu max plus ms upon mu max plus ms upon mu max, and this x naught will come here. This x naught will multiply to the whole equation. Okay, so tb will come here, mu max ln 1 plus s naught minus sf. So mu max will be cancelled out. This and this becomes 0. Okay, so what will remain behind? 1 upon yxs and the x naught here. So tb equals to 1 upon mu max ln yx will come here, then this yxs upon x naught s naught minus sf. Okay, this will become the whole equation. This will the time that will be required, and this is the equation for substrate utilization. Okay, after that what we will do the equation for product formation. Now in this equation the consumption will be 0 and the rate of generation will become, let me write, m will become dpv upon dt, because we have to calculate the product formation. rc will be 0, rg will be qpv, sorry, rpv. It will be rpv, I'm really sorry, this will be rpv. So let's equal this. We will be cancel out where rp is qpx. So let me write qp in the value of x we have determined previously, x naught e to the power mu max tb. This will become here, and 1 upon mu max ln 1 plus pf upon p initial. So this qp will be multiplied into x naught. Okay, this will become the whole equation. From this equation we can determine the product utilization. So this was the end of the video. I hope you like this video, so kindly share this video with your friends, and sorry for the mishandling of this camera, and I have spoken the equations so you can write on your notebooks. Okay, don't follow this writing, then I'm very sorry about my writing. So let's see what will be the response. Okay, I will make videos on molecular biology also, and thank you for all. Thank you very much.